yes, we are live and we can start.
Okay, so good afternoon, everybody. So we start the day 2 session 2, the last session of the conference. So with this, this session, so talks are, so first talk will be by Dr. Tusar Beuria. So let me introduce him by telling something about him at least. So Dr. Tusar Beuria obtained his PhD degree from School of Biosciences and Bioengineering at IIT Bombay. After that, he worked as a postdoctoral fellow at Department of Microbiology and Molecular Genetics, University of Texas Health Science Center at Houston, USA. He has good expertise in the field of biochemistry, molecular biology, and microbiology. Currently, he is working as a scientist at Infectious Disease Biology Group, ILS, Bhubaneswar, Odisha. His group was to understand how the cell division machinery works in bacteria. Interestingly, bacterial cell division is never been targeted for development of antibiotics. His main focus is to understand the mechanism of bacterial cell division and target to develop novel antibiotics. Recently, his group has standardized a high throughput screen to identify cell division targeting small molecules from large chemical libraries. His work has been resulted in several scientific publications and patents. So I welcome Dr. Beuria so to please deliver your talk so that we'll be learning more about your research and we'll have a nice understanding so on the conference as the theme is concerned. So Dr. Beuria, please.
So thank you, Vinod, for a kind introduction. So first, I would like to thank NIT and specifically Sasmita for inviting me to deliver a talk here. So that's the thing. So how to share the slide here?
So you can, there is an option, green option in the below, and you can just click on that and
share screen.
Yes, share screen. Yes.
Okay. So it's shared now?
Yeah, visible.
Visible. Okay.
Yeah.
So Vinod have already introduced me. So I am Dr. Tusar Beuria from Institute of Life Sciences, and my group works to understand the bacterial division and if we can actually target it to develop novel antibiotic. Now here if we look at like how infectious disease can cause havoc. So this is actually old data by WHO in 2013. So what it says, like these are the number of years lost due to ill health, disability or early death for 1 lakh people. All over the world it is there. So if you look at India, so we are losing almost 4,000 to 5,000 working years for 1 lakh people. That's a huge amount of time we are losing. Now this is a recent data actually. It is showing up to 2017. So if we look at the communicable disease, it is actually the death occurs due to communicable disease is almost 25% of the total death happening in the worldwide. Okay, so it's actually one of the major cause of the death. And in those infectious disease or communicable disease, if we look at top 10, there are almost 5 of them are bacterial disease. Okay, and there is tuberculosis is one of the highest killer among the bacterial disease. So that is here in the 4th position. Okay, now this is a recent data by WHO in 2018. So you can see here like the top 10 global cause for death, that is out of this top 10, 3 are due to infectious disease. And you can see tuberculosis is the number 10. Okay. Now that is actually that is not a major concern. The major concern is if you look at drug discovery, antibiotic discovery, there is an antibiotic discovery void after 1990. So no new antibiotics has been discovered. Of course, there are a couple of antibiotics discovered, but these are generally the modification. The reason for that is no pharma is interested to make any new antibiotic, because it's directly depends on the finance, how much you are putting. If you do a research, any pharma, if they are doing a research on uh, antibiotics, they have to spend a lot. But antibiotics generally, people, they don't take for long time. It's for 5 days or 7 days course. Like other lifestyle disease like blood pressure or diabetes, so you have to take medicine every day for 30-40 years. There where major pharma are focusing. And that is not the only problem, that no new antibiotics are coming. But once it is coming, you can see here, next couple of years bacteria is getting resistance to it. So that means uh, antibacterial resistance is also adding to novel antibiotic discovery. Means in both way, we are not generating any new or novel antibiotics. Apart from that, whatever we have, we are getting resistance. All the bacteria are getting resistance to all those available antibiotics. Okay, and this antimicrobial resistance or antibiotic resistance is not a problem to any single country. It is a global problem. You can see here that all the continents has been equally suffered due to this, and it has actually a lot of financial implication apart from the death. Even Antarctica, people have actually identified antibiotic resistance bacteria in Antarctica. Okay. Now, what is the remedy for that? What we can do? So, one of the reason why we are working on the bacterial division is never been fully understood. It's still very new topic for the bacteriologist. There is it has been discovered like uh, bacteria division is discovered in 90s, but no people were working with this. So it only took up after like 2005 or so, like many research groups started working on this. So here in my presentation, I will show that why this bacterial division is an attractive target for novel antibiotic discovery. So before that, I'll just tell a brief introduction about the bacterial division. So these are the new daughter cells which grows and the DNA segregation take place, and a ring forms at the middle. This ring is known as the G ring. This G ring actually constricts and then divide the cell into two identical daughter cells. Okay, so this ring looks simple, but it's a quite complex ring. There are at least 30 proteins, some more than 30 proteins. They are known to be the part of this G ring, and this G ring also coordinate with other cellular processes like uh, peptide glycan synthesis, that is cellular formation, cellular breakage, motility, DNA segregation. All these independent processes, it has to coordinate. It has to coordinate, and how this coordination happens, that is still not fully understood. We know a little bit here and there, but there are a lot of things there to discover. And in this bacterial cell division proteins, which we also call it as divisome complex, FTSG is one of the main proteins. So FTSG first gets recruited to the mid cell with the help of GPA, FTSA, then it other proteins, they are either enhancers or inhibitors of the uh, divisome complex. They gets recruited and they form the ring, and this ring formation uh, is quite dynamic process. That means this ring has to form, then it gets stabilized, then it should generate the power to constrict the cell wall. Then that constriction process is quite dynamic in nature. Okay, we still don't understand fully how this process is happening. And this FTSG is a very well-known bacterial homologue of tubulin. So if you look at the structure of FTSG and tubulin, they are quite highly similar. There are 60-70% similarity are there. And apart from the structural similarity, there are almost 18 to 20% sequence similarities are there. And there are specific sites where both FTSG and tubulin are very similar. And they also have similar functions, like they bind to GTP and they assemble in the presence of GTP and disassemble when they make the GTP hydrolyze. So many of the properties between FTSG and tubulin are quite similar. And if we want to know like how this FTSG is helping the formation of this ring, so here you can see that these are the FTSG monomers. They in the presence of GTP, it forms the single protofilament. You can see at that time this ring formation will start. Then GPA, JAPA are the enhancers. There are other enhancers also recently discovered. Then EGRA, MIN-CD, FTSA, there are certain inhibitors of FTSG bundle formation. So all these proteins, they make sure that formation and uh, uh, inhibition of the ring take place simultaneously, so that the ring is quite dynamic in nature. So this ring gets stabilized, and after that, that will constrict and divide the cell into two identical daughter cells. Okay, now here this is a confocal image of the G ring. It has to form at the middle. So how this ring formation taking place at the middle? So there are actually technically, if you take only FTSG can actually polymerize, and it can form ring all over the cells technically, but it does not happen in the cell because there are two different systems. One is nuclear occlusion system, which makes sure that ring formation does not take over the nucleus. So there are supposed to be ring formation, but over the nucleus there is no ring formation, because this NOC protein, they inhibit FTSG polymerization over the nucleus. And another system is the MIN system, which is again another inhibitor, which makes sure no ring formation takes place at the poles. So these two systems make sure that there is no ring formation over the nucleus or over the poles, so it can form only at the mid cell. Okay, now the question is why we need to study the bacterial division. Okay, this figure I have already explained. Now this, if this G ring forms normally, we'll see that lot of bacteria divides, new daughter cell forms, and there will be lot of bacteria. And as we know that bacteria is one among those fastest growing organism. It's like, for example, E. coli, it takes 20 to 30 minutes to get double. In one day time, from one bacteria can become several million or several billion bacteria. But if we can inhibit this ring formation, then what will happen? Cells cannot divide. So nucleate segregation will take place, but the cell will not able to divide. So all these filamented cells are single bacteria. So technically that number, they cannot increase. And after certain time, what happens? Now, those filamented bacteria, they lyse. Okay. Then how this FTSG plays a role? So FTSG, these are the electron microscopy images of FTSG polymers. FTSG normally forms very good polymers if you look them under the electron microscope. And in the presence of inhibitors, those polymers will form very small polymers. Okay, or they will disassemble. And in the presence of enhancer, they will form bundles, like lot of bundles of polymers. In both the cases, as I mentioned earlier, that G ring is quite dynamic in nature. That means it has to form, then it has to disassemble. So any of the process is inhibited, then that cell division will hamper, will not bacteria will not able to divide. So here I'll give you an example, like in the presence of inhibitors. So these two molecules actually we identified in our laboratory. One is doxorubicin, that is inhibitor of the FTSG assembly. You can see that is the outer membrane which is staining by FM 464, and there is no ring formation in the middle. So there is lot of filamented cells without any FTSG ring over there. And in the presence of enhancer, so what enhancer does, they enhances the division complex. That means division complex will form, but they will not depolymerize. So you can see this is actually astronam. This is one of our work which we have not yet published. So here you can see that there is a lot of filamented cell, but having lot of Z rings over there. Okay, so that means in both the cases, cells will be filamented. One in with the lot of Z rings, one in the absence of G rings. Sorry, this is with a lot of G rings, and this is without G ring. But both the cases, the cell will die. Okay, and these are three different drug binding sites. If you look into the literature, there are at least 150 to 200 literature are there which are like describing about different small molecules, a group of small molecules which can interact with FTSG. So they are generally interacting with three different sites. One is GTP binding site, one is T7 loop, and one is this part is in interdomain cleft. Okay, so there are three different binding sites. They are characterized within the FTSG, and I'll tell about that later. So before that, I'll come to like one of the screen what we have done in our laboratory. So what we did using like microbiology, microscopy, docking, and the biochemical assays, we screened one FDA approved drug library. Then we found that two molecules, those two I have already told you in the previous slide, that doxorubicin and the astronam, they interact with FTSG, and they actually either enhance or inhibit the G ring formation. One of the molecule what we have already published, that is doxorubicin. So what we saw that you can see that clear filamentation that is happening in the presence of doxorubicin, and I'm not describing that in very much detail, but we then characterized the doxorubicin binding site. We identified the site, and that site is a different binding site than the GTP binding site. And here you can see that two different sites are there. One is GTP binding site, and the other one is doxorubicin site. And we made mutation, and we showed that this if we make mutation in this doxorubicin binding site, which is S10 helix, that inhibits the binding of doxorubicin to FTSG. And here you can see this is the GTP binding site, and this is the doxorubicin binding site. And this site is present in many different species of the bacteria. It's not like that it is only present in the FTSG from E. coli, but like MTB it is there, B. subtilis it is there, Staph aureus it is there. So that site is quite conserved. So this site can be in the future can be targeted for the development of new drug molecules. Okay, now this is one part of my work, but today I'll tell something, a success story about some compilation of works from different groups, like how it started from a very low efficacy molecule, uh, how that molecule converted into a very highly effective molecule, and the target was FTSG. So this is actually the molecule is 3 MBA, or that is 3-methoxy benzamide. So here in 1999, so that is 20 years back, Kawamura group from Japan, so they actually screened certain molecule, and they found this 3 MBA. And what they found that before that, like 3 MBA was a known inhibitor for ADP ribosyl transferase, and what they found that it is making filamentation. These are the normal cell. In the presence of 3 MBA, lot of filaments are there, and they did biochemical characterization, they found that 3-MBA is interacting with FTSG. And here, I don't know if it is okay. Okay, so then if you look at the efficacy, the MIC value is 2000 microgram per ml. So that is 2 milligram per ml, which is very high concentration that never can be used as a drug. But what they have that time showed that it can bind to FTSC and it has effect on bacterial division. Even the effectiveness is very less. Then in 2008, David Hedden and Neil Stroke group, actually they did a modification. They actually made or chemically modified this compound. They screened over 500 analogs, and they found this addition of this group increased the efficacy of the drug by several thousand times. That it was 2000 microgram per ml, but what they found with this modification, the MIC goes to 0.5 to 1 microgram per ml. Okay, and this molecule is now very much active molecule. Okay, now later it was found that this molecule goes to the interdomain cleft of FTSC. Okay, but it has like very poor performance. It is like the pharmaceutical and pharmacokinetic properties are very poor for this molecule. And this molecule, that's why it could not administer orally. It was not very much effective. Then Daniel Pilch group, this group is from New Jersey, that is Rutgers Robert Wood Johnson Medical School. So they found another molecule, that is TXY 436. Actually, this is the group which have done a lot of chemistry on this FTSG targeted molecules. So what they found, they just did a modification into the this PC19073. Here with this modification, what they found, that the solubility of this molecule is 100-fold more than PC190723. Okay, and MIC value is kind of similar, 0.5 to 1 microgram per ml. And now, as the solubility of this molecule is very high, this is very much suitable for the oral administration. And orally, they have also checked that orally bioavailability of this molecule is quite high in a mouse model. And similarly, they have also checked its effectiveness towards methicillin sensitive as well as methicillin resistant staph aureus, and towards both it is quite effective. Okay, and I'll tell here that this TXY 436, this is actually a prodrug. So what happens once it is administered, then it will change its chemistry from TXY 436 to PC190723, and this has a like half life of 18 minutes. Okay, then the same group, they also did another modification, that is 1-methyl piperidine. They added this group here, and what they found, this is even much better than the previous molecule. That is solubility wise, it is 143 times more soluble than that, and it is also orally. Uh, it was actually suitable for both oral and intravenous administration. But there is a problem that it suffered from suboptimal pharmacokinetic, and the requirement that's why high dose was required, which was not advisable things for this. Okay, and here now the T half in the previous molecule, the T half of this one of the prodrug, like this molecule to PC190723 was 8 hours here. So previously it was 18 minutes, but that is in Staphylococcus growth media, but in mouse serum it is only 3 minutes. Okay. The same group in 2015. So if you look at their publications, every 2 years they modified this molecule, they made another modification here, that in the chlorine group, this chlorine group, the if you look at here, this chlorine group is here. So they modified this chlorine group to CF3 group, and now okay, now that makes it like metabolically quite stable, and it is quite efficient against Staph aureus strain, that is resistance towards vancomycin, daptomycin, or linezolid. Okay, now this is also a pro drug. That means this drug will convert it into TXS707. It is not a pro drug for PC190723, but it's a pro drug for TXS707. Okay, and TXS707, in the same paper they have showed that it is quite effective molecule against many different resistant bacteria. Okay, now this product is actually 6.5 fold longer half-life. That means it can stay longer in the blood, and it can continuously supply the drug molecule for longer time to the blood, and it also had threefold greater oral bioavailability. Okay, but it faced another problem, that when this drug was administered in the mouse, there are two strains of the FTSG which get resistant to this drug. That is, when there is a mutation at glycine 196 position or glycine 193 position, then this Staph aureus becomes resistant to this both the drugs. But later they identified the mechanism. What they found, that this resistance is mainly due to the steric hindrance. Okay, the same group, what they did in the 2017, they have published this data. So here, actually, I'm sorry for this figure. I had to made in the similar way, that's why I have to just flip it. So what they did, they have modified this part of the drug. So they made it flexible. If you look at the previous molecule, you can see that this is actually joined, but here they made it flexible, and due to that flexibility, that steric hindrance which was supposed to inhibit the uh, uh, which was actually creating the drug resistance, that did not happen for this molecule. And here they have also shown the mechanism how this molecule can flip, and if there is a mutation in those G196 or 226 position, then still it can actually inhibit the growth of that bacteria. So what they have done, they made the ring flexible, and MIC was even better, that is 0.12 microgram per ml against sensitive strain. And when this MIC value against the resistant mutant was 64 for the previous molecule, now it was become 1 microgram per ml. Okay, so that become much better in presence of this. Okay, now this TXS709, it was actually taken up by one company, that is Taxis Pharmaceutical. So later what they proved, that TXS709 is also synergistic with available antibiotic, that is Cefdinir, and they showed that 50% of the 50% concentration of the normal dose of Cefdinir when given along with the TXS709, that is quite effective to kill the bacteria. Okay, and later they have again taken this, and now this drug they have applied for phase 1 human safety clinical trial. So with this, I'll just summarize my talk, that cell division in bacteria is never been exploited to develop antibiotics. Now people are doing it. That's why the understanding of bacterial division will help us to understand one of the most potent antibacterial target. And FTSG, a widely conserved protein in prokaryote, is a potential target for development of antibacterial compound. And this, my work was mainly done by two students. Pragyam was a postdoc. Ashok was the PhD student, who is now doing his postdocing at University of Florida. And then Ansuman Dixit is one of my colleague who actually helped us to do all the uh, uh, computational work. And we got funding from ILS, from DBT, uh, extramural funding, and also the SURF funding. So with this, I'll actually conclude my talk. Thank you. So any questions?
So I think so this forum is open for discussion now. So please, you can ask the question directly to Dr. Beuria. Anybody?
So
At least from this Zoom platform.
Zoom platform, yes. I think anybody who wants to.
Dr. Binod, can I ask one question?
Please go ahead.
Okay. Dr. Tushar, it was a very nice talk actually.
Thank you.
My question is, and when we are making
So,
like
Dr. Negi, sorry, I'll just pause you
Yeah.
for a moment. I think Dr. Beuria can remove his slide, like can withdraw his slides.
Stop sharing.
Yes, stop sharing.
Okay.
And we can see. Yes, please.
okay. So can I go ahead now?
Please.
Yes, from my side, yes, or I think.
Okay, fine. So as this protein is like a well-worked protein, and you have targeted specifically this species. So can this drug what you have worked be used for any organism, or it is specific to the organism you're dealing with, or because of the conservation of this protein? No? So, or have you tried in any other organism?
So organism means different bacteria you are talking
Right,
about.
right. I meant
Yeah.
that only. Huh?
So, yeah. So it is actually FTSG is quite conserved among all the bacteria.
Right. Mm-hmm.
So it will be if you are targeting FTSG, generally it should be uh, broad spectrum antibiotic. Okay. So generally it should target most of the bacteria. It should,
Right.
but actually it depends on the metabolism of that drug. So sometimes it is gram positive. It is like more effective towards gram positive. Sometimes it is more effective towards gram negative, although it targets FTSG. And there are
Okay,
certain
yeah, please go
differences.
ahead.
There are certain differences in gram positive and gram negative bacteria, so which can be specifically targeted.
So my concern is because when we are using this drug, wouldn't it be as effective as it is against pathogen, against our microbiota, which is in our gut?
Mhm. Yes, that I understood. So that is one of the question we get, but actually that can be targeted. That can be targeted, and any antibiotic cannot be like very specific. If you look at
Right.
the antibiotics, so that plus minus things will be there. But another thing for the antibiotic, mainly the course of the antibiotic is for 3 to 5 days or 7 days. So
Yeah, and then we come back with the probiotics to bring back the biota.
so a lot of things will be there, but can be targeted, because if you want to target a particular organ, so you can actually modify this drug, like where to metabolize.
Okay.
Okay. So how to
So maybe the route, right, and the route of administration could be changed. Like instead of going with the oral, if some other route is utilized, that may be more effective than going with the oral.
Yeah, generally intravenous is more effective than oral,
Oral, right.
because oral you need a higher concentration or a higher dose of that particular antibiotic.
Correct. Okay. Okay, sir. Thank you. Thank you very much.
So I think we have 2 to 3 questions coming up from YouTube channel. So if I have the permission, should I ask?
Please go ahead.
So Prabhat Kumar Swani is asking how to increase the solubility of TXY436 as per your drug delivery process.
TXY.
So he is asking about the solubility of this drug, TXY, whatever particular name he has given. So how you are going to increase the solubility of this drug as per the process you are following?
Okay, so one more thing. So I think all these molecules, like the last part of the presentation was not my work. Okay, like all the TXA work is mainly what I presented, like how the 3-MBA was converted gradually over the years and made a prodrug. Okay, so is mainly many chemists are involved in that part. So what they did, they made lot of analogs and identified which analog is more soluble. So there are many analogs. Like for example, they add CF3 group. CF3 group is mainly one of the thing is for solubility, and also it is less effective towards the metabolite. So here one of the metabolite is cytochrome 450, which can actually degrade this drug. But his question is specifically about the solubility, right?
Yes.
So you can look this part. So TXA 436. TXA 436 is the first molecule which was where they have actually added that amine group here along with another amine group, which made it more soluble. Actually, chemistry people can tell it little better. If you
I think
look at
solubility can be enhanced by changing the pH, changing the temperature, and as well as adding some chemical
adding the
substance. So
adding
they
the
interfere with the molecular bondings and things can be solubilized in proper conditions. So you have to make it a trial and error to see which solvent and which things are actually appropriately helping in dissolving, restoring the chemical composition and activity of the drug molecule.
they are actually they have added polar group to make it more soluble. There are a lot of I actually choose certain very specific molecules, but if you look at that literature, they are gone through like lot of polar derivative. So polar derivative will make it more soluble. That's how they came up with it.
Another question is, like, is there any specific cause of methoxy group in 3 position in your 3-MBA?
Methoxy group in 3-MBA specific cause means so
So maybe he is asking about the structure, like if it is like that, or you have made any modifications with some intentions. So he's basically talking about the position, grouping methoxy group position in 3rd position of third
that
3-MBA.
molecule again we are actually not working with those molecules, but that molecule was identified long back. It is something else. People were screening that, and they found that 3-MBA is effective, but 3-MBA was not that effective. So methoxy group has any role exactly? I cannot tell that.
So Ankita Solankar, she's asking one generalized question like using of TXA 707 as a drug to cure any disease like are we using is it in current use?
No. That I told that 707 is a drug molecule and its prodrug is 709. That means 709 is the drug molecule which it will enter to the body, then it will convert it into 707. And this part is a company that is Taxis company. They have applied for FDA approval for the phase 1 clinical trial. So it is not in use.
Okay, still in under validation process. Okay, so I think I have taken most of the questions. Yes. So
Anyone from the Zoom platform has any question? Okay, good. So with this, then I think we finished early, right? Early ahead of as per the scheduled time. So Dr. Beuria. It's a very nice talk. So people learned. And if anybody has also a question, so they can email so Dr. Sasmita Nayak madam, and she will be in touch with the presenter, and the answers can be also given. Yes.
Yes,
So
sure, sir.
then with this, we thank very much Dr. Beuria for his nice talk. So I think so most people understood the stuff correctly and with few corrections, those who asked the question. So then with this, maybe we can proceed to the next talk.
Okay.
Bishmita madam.
Yes, sir. I think
So
we can.
thank you, Vinod,
Thank,
and thank you.
thank you, sir. Thank you, sir.
Thank you,
Thank you
sir.
so much.
It's a heartfelt thank you, and thank you for such a like you could manage with such a short notice. So we are very
Yeah.
delighted to have you here because I had never interacted, never been face to face. I have seen you. We learned a lot, and it was really nice interacting, learning from you. So thank you so much.
Thank you.
So should we start the next session,
Yes,
Bishmita madam?
yes, sir. Please go ahead.
Indranil sir is there. Indranil sir.
Hello.
Hello sir.
Hello. It's nice to hear you.
I cannot
So
see you,
we
sir.
cannot
Just a
see
second,
you.
sir.
Yes, sir.
Hello, sir.
Hi.
So
Hello. Hello, Dr. Negi.
Hello, sir. Hello. How are you, sir?
I'm fine.
It's
Hello,
been,
Vishmita.
Hello,
how
hello.
are things there,
Nice
sir?
to see you after such a
Yeah,
long
yeah.
time.
It's a very nice experience seeing all of you again together.
Hello, sir.
yes,
I
sir.
know, I know.
Hello, this is
So I was
Vijay.
looking forward.
Hello, Bijesh.
How are you?
I'm fine. How are you?
I am good. Nice to see you after long.
Same here.
Uh,
See, I'm walking and listening to you. I have another meeting to attend. I'm just rushing there only, sir. Okay. Please
Okay,
go
okay.
ahead.
No
I'll.
problem, ma'am.
okay,
Please.
so we'll listen to you.
Okay.
Sure.
Okay, so with this, maybe we can proceed to the second talk of the day 2, session 2. So the talk will be delivered by Dr. Indranil Banerjee. He was actually here, almost here in the our biotechnology and the biomedical department in IIT Rourkela. But lately he shifted to IIT Jodhpur, and he is working as associate professor over there. And with the specialization, tissue engineering, drug delivery, regenerative medicine, biomicrofluidics, then theranostic system, and as per the academic profile is concerned, he got the MTech so in pharmaceutical and fine chemical technology from University of Calcutta, followed by PhD from Department of Biotechnology, IIT Kharagpur. And also he has been invited as guest scientist, Max Planck Institute of Intelligent System, Stuttgart, Germany. And he has published 70 international articles, and the total citation in the Google Scholar as we can find is 1897 with H index 25 and I10 index 49, and cumulative impact is 200. And he also edited 4 books, and also has been engaged also in addition to his regular academic and research job. He is also associate editor to Frontiers in Medical Technology. He has a lot of expertise actually. So and he is very much expert in his topic. And but for today we will be learning from his experience on the title cell derived membrane vesicles as novel therapeutic cargo. And so I wish Dr. Banerjee to please deliver his talk. And any question you can keep posting in this chat forum, as well as we are also live in the YouTube. Please keep posting the question as we go along, because sometimes we keep forgetting question, right? Towards the end, okay, what I will ask. Okay, so if that will be there, then we'll be asking the question on behalf of you, and it will be so nice also. And in addition, the Zoom platform, anybody can also directly discuss with the speaker. So Dr. Banerjee, please.
Thank you, Dr. Binod, for this nice introduction. So, first of all, I would like to know, am I clearly audible?
Yes.
Yes.
Okay, okay, thank you. So let me first share the slides, and then I will start talking about, okay, just a few seconds.
Please go ahead.
Hello?
Yes.
Yes.
Yeah, is my
Audible?
slide visible?
Not
It
yet.
is still in the process of started
Need to
sharing.
double click to make to screen full screen mode.
I went to full screen mode only.
Still we are
Okay,
in
resume share.
black screen.
Just a second.
Yeah.
Is it coming now?
Not yet, sir.
Showing shared. Maybe some delay.
Yeah, quite possible.
So it is actually showing that you have started screen sharing, but it has not yet come.
That is the thing. I think there is some network issue.
Maybe if you go to the next slide, this first slide will come.
See?
No, sir.
Just a second. Actually, Zoom is, we generally do most of these things in Google Meet or WebEx. So,
Right.
but I don't know why it is taking so much time. It is still not been shared, no?
No, sir. So can you take out and again try it again?
Just a second. Stop share.
Yes, sir.
Again.
Now we can see you.
Yeah. Now share screen.
Yes.
Yes.
Is it coming
Now it has come. Yes.
Yes, sir.
Okay. So there was a delay in I believe.
Yeah.
So you can make it a full
Yeah.
screen.
I am doing that. Just a second. Is it okay?
Yes. Very perfect.
Okay, fine. So thank you, Dr. Bishmita, and all my friends there at NIT Rourkela. So it is a nice experience. Then I still feel a very strong attachment with the NIT Rourkela. For the common audience who are listening this talk, so let me explain that point. So actually, I was on faculty at NIT Rourkela. I served that institute for last for 8 years, and after that, I moved to IIT Jodhpur. But I had a very close association with all these friends who are actually right now attending this conference and its online platform. So seeing all of them is a very nostalgic experience. And I'm feeling really very good, and I'm grateful to Dr. Bishmita for giving me an opportunity to present here. So with these words, I would like to proceed. So may I?
Yes, sir. Sure. Yes, please.
Thank you. So today I will be giving a talk on the cell-derived membrane vesicles as novel therapeutic cargo. Now, before going into the details of this talk, just let me brief a bit about my own research. So I work in a very interdisciplinary domain. So that domains have small subdomains that includes therapeutics, theranostics, tissue engineering, drug delivery, mechanotransduction, biomicrofluidics. Now all are in their own at a very big topic. So it is not that I work in on every aspects of this individual topic, individual domain. So I work in specified areas, mostly related to that drug delivery and bone tissue engineering and angiogenesis. These are the common areas, and I pick up some of these relevant technologies and issues pertaining to these topics. So today, as I said that I'm going to deliver the talk on the cell-derived membrane vesicles as therapeutic cargo. Now we can start with that point that in the last 50 years, people observe something that from the cells, it often sheds some particles. So particles of various dimensions. Sometimes they observe it is in micro dimension, sometimes they observe it in nano dimensions. In earlier days, people generally considered that this kind of, say, the particles, what they are observing when they are dealing with the cells, both in vitro or in vivo, they are basically a kind of debris that coming out of the cells. But the later phase, people realize that they are not at all a debris, but they are a very important bioactive components that are present in vivo and in vitro, both the situation, and serves dynamic purposes and which are very, very essential for various kind of cellular phenomena. So that cells as a living entity, it generally secretes 4 different kind of vesicles. So these vesicles, if you go in and say layman's word, if we want to tell that, they are vesicles because they are enclosed entity. They are actually, there are certain kind of cell membranes, that part of the cell membranes or lipid membranes that that hold certain inner components, and these vesicles have different size and different function. So from the cells, people generally have 4 different kind of vesicles. From the naturally, if I here you can follow the cursor, that one is very common nowadays, we all have heard about it, that is known as an exosomes. Another one is an apoptotic body, which generally obtained from that apoptotic cells. And another one is in microvesicles. These are obtained from the naturally say cells in a natural condition. So these 3 are the different kind of vesicles that a cell generally produce. And there is another kind, GPMV. This is giant plasma membrane vesicles. Today I will devote most of part of my talk on this particular topic, this GPMV, giant plasma membrane vesicles. But before reaching to that topic, we need to have an idea about what these different kind of vesicles are. Now, if you look into this, here is a small table which summarizes the properties of different kind of vesicles. The first one is an exosomes. So if you look into this, it is a typically nanoparticles or nanovesicles. So their range is generally 40 to 100 nanometer. So here, these exosomes, cells in normal condition keep secreting these exosomes. And these exosomes they contains a lot of bioactive molecules, including small molecules, then microRNA. These kind of bioactive molecules make these exosomes as a very, very effective communicators between the cells. So if you look into the literature nowadays, you'll find that exosomes have been a point of interest for various kind of research, especially in that cancer and cancer vaccines. And then there is a microvesicles. These microvesicles, if you look into this, it is a very heterogeneous, it encompasses a heterogeneous populations of vesicles. So it could be from starting from 20 nanometer to 1000 nanometers, that could be in dimensions. And then there is an apoptotic bodies, which are generally of a larger size of 1 micrometer to 5 micrometer. So all these kind of vesicles, they contains different kind of bioactive molecules. Now in that schematic, you can see that the apoptotic body is basically when a cell is dying, from there it has been produced. And in the other cases, exosome and microvesicles, they produced from live cells most of the time in healthy conditions. And when they produce, they contains a lot of bioactive component, as I mentioned just now. So it could be, and their plasma membrane compositions have a very close resemblance with that cell's own membrane. So altogether, because of this kind of vesicles, as they contain some bioactive molecules and their plasma membranes are having the native compositions, they have been a point of interest for research in the recent years. Now here's some of the images, you can see this. The first one is that cancer cell. Here it is secreting these exosomes. Now, just for that sake of informations, I can tell you that this kind of exosomes have often been used for the intercellular communication. So one cancer cell secretes some exosomes, and that exosomes been taken up by either another cancer cells or some other cells present in that cancer microenvironment. And by sending this or by communicating through these exosomes, cancer cells send a very important signals to the neighbor cells and make its own microenvironment hospitable for its future growth. So that's one kind of use of these exosomes in the natural or pathobiology of cancer. So here you can see this, Here you can see this in the next image. So a killer T lymphocyte orange inducing a cancer cells to undergo apoptosis through this help of this, and here is that apoptotic bodies that cancer cells is producing. Now in the third image, this black and white one, here you can see that the platelets are secreting microparticles. And the last one, as I said, we'll discuss in details about this. This one is a giant plasma membrane vesicle. So you can see this bigger or brighter green is in one cell, and from this one cell you got another vesicles of almost similar size. So that's why they are generally termed as a giant plasma membrane vesicle. So these are I'm giving these things just to have an idea how this different kind of plasma membrane vesicle looks like. So now what are this function of this cell-derived membrane vesicles? So they have a diverse role, as I mentioned earlier. They are used in inflammation and immune responses. They help in antigen presentation. Nowadays, many kind of vaccines people are trying to make on the basis of this cell-derived membrane vesicles. They are a very excellent immunomodulators. They can change the balance of this immunological reactions to any directions. And it has been observed that some kind of this membrane-derived vesicles have a unique role in tissue regeneration. So it is often been observed that membrane vesicles secreted by stem cells, especially mesenchymal stem cells or hematopoietic stem cells, induces or certain kind of regenerative response inside the tissue through cell-cell communication. And just like regeneration, it also helps in repair as well. And it is a means of different kind of communication, this kind of exosomes. And you will be surprised to know that this exosome or this membrane vesicle communication, so often cell use to trace the path of migration. So it is just like our old days fairy tales, Hansel and Gretel's. So where they keep small pieces of bread to keep a track through which they have moved. So when a cell migrate, so it keeps delivering certain kind of exosomes on its path, and the later phase another cells can follow the path and taking these things as a clue to which directions they need to move. So these are the very unique features of this kind of membrane vesicles. And in the other way, it can induce senescence as well. It can influence the proliferation and differentiation, and it can help in cancer progression as well as metastasis. So these are the general features of this function of cell-derived membrane vesicle. Now, as I said that all these things, how these are coming into our, present talk. So what is their relevance in that drug delivery? Now, here you can see that these membrane-derived vesicles, they are their typical compositions. EV is that extracellular vesicles, what we are talking about membrane vesicles. So these EVs, their surface and membrane proteins are so specific that their interactions can be a specific kind. That what I'm trying to say is, so a particular cells when secrete a kind of membrane vesicles, that membrane vesicles have an affinity toward another kind of cells, and that is very specific. It is not that it can randomly go and interact with any other cells. So it has certain kind of specificity towards another kind of cell. So these things are often found less immunogenic because the compositions is such that it is derived from these cells and their membrane compositions remain as of the native cells. And especially if you get it from that autologous cells, there won't be any questions of immunogenicity. And then it can bypass the complement activations and interactions with the coagulation factor. These two are very, very key parameters, because if you want to use this kind of membrane vesicles as a drug delivery cargo, so when you administer these vesicles inside the body, they won't react with the complement or interact with the coagulation factor. As a result, their persistence in that blood will remain for a longer time period. So that desired outcome, there is a possibility that you can get a more desired outcome. And then, as I mentioned earlier, they recognize their target cells. as they can recognize the target cells, so there is a reduction in the off-target effects. Now, when you use this one, so this kind of membrane vesicles, so nowadays there are two different kind of drug loading approaches are used. So one is known as exogenous and another one is an endogenous approach. So what is the endogenous approach? In endogenous approach, we first tune the cell. Suppose we want to load a particular kind of microRNA. I want to deliver a microRNA to another cell. So first what people need to do is that they need to engineer a cell in which this microRNA be produced, and then they get this microRNA packaged in these exosomes or any cell, this cell-derived microvesicles, and that can be delivered to another cell. So same thing is true for, it can go with any kind of membrane protein, and the membrane protein can bound with this and that can be delivered. So here why it is endogenous, because the cells, you are loading the particular your molecule of interest, a drug, a therapeutic molecules, and nucleotides, whatever. So you are loading it at the beginning where this particles is being synthesized. And what is that exogenous loading? Exogenous loading is that you are taking any cells and this corresponding cell-derived vesicles, and then in the cell-derived vesicles you collect it, then you can load your drug or you can modify the surface, and then finally you can administer it or use it for delivering that particular drug. So these are the general approach of playing with the membrane-derived vesicles. Now, if I take a small pause here, so my point is, so far what I'm trying to say is, from the cell, we can get some membrane-derived vesicles of different size. It could be apoptotic body, it could be exosomes, it could be microvesicles. These can be derived from the cells. Now, in this particular vesicles, we can load our drug either exogenously or endogenously, and these things can be used to deliver the particular drug or therapeutic molecules. So this is so far that outline of this part of the talk. Now question is, whatever we are talking here, all these three kind of vesicles, they are naturally synthesized vesicle. So it is not that we are tampering their synthesis. So eventually, when they are being synthesized, they are carrying the native properties or any characteristics of the cells at that particular, so which is corresponding to the physiology of the cells at this particular moment. So what I'm trying to say is, suppose if you want to collect this kind of vesicles from bodies, some patient sample or some donor sample, one day you will get these vesicles of one kind, and the next day you may not get the same kind of vesicles because of the change in the physiology of that particular cells of that from the donor. So that kind of variations is always associated with these naturally derived particles from the cells, because we are not in a positions to engineer the synthesis. So question is that, so is it a true bottleneck, or do we have any other options to play with? So here comes the next set. So now, as I mentioned, that the synthesis of these natural extracellular vesicles depends on the state of the cells, and we do not have a control over the chemical composition of it. So it is associated with poor reproducibility. So you can reliability of this process is very less. And people always question, so whether today I'm getting this result, will I get the same result tomorrow if I follow the same protocol? And answer is definitely uncertain. So here comes the next point, which is known as a GPMV. So giant plasma membrane vesicle. So what is that? Now GPMV, before going into the slide, let me tell that GPMV is in similarly cell-derived membrane vesicles. But here the point is, it is not been synthesized by the cell automatically. Here we are inducing the synthesis, And depending upon our choice of chemicals or our choice of external cues, we can tune the synthesis of this giant plasma membrane vesicle. So here is the scheme. So what is the scheme? Please have a look. So this is a normal cell. So in this cell, so it contains, its membrane contains all the receptors, surface protein, ion channel, everything. Now, if I add a particular kind of chemicals or other external cues, which are generally being termed as the vesiculating agent. So then what happens? This vesiculating agent will compel the membrane to make certain kind of blebs. And after formation of these blebs get detached from the cells in the form of a vesicle. This vesicle is termed as a giant plasma membrane vesicle. And why it is different than the normal plasma membrane vesicle? Because of its size. Its size is relatively of higher side. So generally, these have a size of 1 to 15 micrometer. Now, all these vesicles are fluid filled. So bilayer cell membrane vesicles. And if you look into this, what I'm trying to point out here, when they got synthesized from the cell, they carry the same surface receptors and surface proteins and ion channels, everything. So technically, they mimic the whole cell membranes and a kind of cellular environment. So with this, now the question comes, can this kind of membrane vesicles be utilized for drug delivery or as a cargo? Now, when we are trying to talk about this, its use as a cargo, we just need to think in two ways. One is that what is actually inside? So this particular portion, as we said, the fluid field, what is there? What is the fluid? And the second point is that can we really manipulate it for our own, to satisfy our purpose? So the point is, this kind of giant plasma membrane vesicle, they generally contains that part of the cytosols. And in the cytosols, it contains a lot of different kind of bioactive molecules. So here, let's have a look into this, what kind of bioactive molecules it can. So these are some data of my own labs. So here, just to give you some idea about this. So we generated these giant plasma membrane vesicles from cells, and then we stain it first to check whether it contains protein or not. We use two different kind of protein markers, non-specific protein markers, not for a specific antibody kind, non-specific protein markers. One is THT stain, another is an qubit. It is a commercial protein marker. We find the signals there. So that means it contains the protein. Then we stained it with an acridine orange and check it as an appropriate wavelength. And there we find that this acridine orange is a marker for this RNA, and we find that the presence of the RNA inside. Similarly, we go with the qubit RNA, we find the signal here as well. Although this is in the image, it is not that clearly visible. I guess that you may not be able to see it, but when we zoom it, we found that the signal is there. But when we check with the hoist and the qubit DNA to DNA specific dyes, we did not find any signal. So that means that this kind of membrane vesicles is devoid of any kind of DNA. So technically it contains RNA, it contains proteins, but it don't contain any DNA. Then we check what is the distribution of kind of this RNA and DNA inside. So we find that this particular kind of membrane vesicles, this whole red one is in one membrane vesicles, where you can see that there is an RNA distribution. And what is very interesting here, that the distribution is a kind of heterogeneous. So that means that a certain inside this whole inside this GPMB, the cytoplasm itself is heterogeneous. So the distribution of this particular di-RNA conjugate is not uniform, and that's why you are getting this kind of heterogeneous distribution. The same is true, but in case of protein, we get the distribution is a little bit more homogeneous in nature. So these three experiments altogether confirm that GPMP contains certain kind of cell-derived bioactive molecules. So one way it satisfies the criteria that we can use it for say any kind of therapeutic purpose. How, that we need to explore, but it can be done. Up to this has been proved. So now I would like to give you an idea what actually, so what actually this GPMB looks like. So far I have just confirmed that these particular vesicles can be used for delivery. So just give me a second to share a video. Is the screen visible?
I think you have reduced the slides, but
Yeah, now I'm just going to share a video.
okay.
Is it visible?
So video has not yet come on the screen.
Okay, I'm requesting a little. Just wait for a while. I think
Yes.
video will be visible.
Yes.
So screen sharing is paused. Why? Okay, sorry. Now, sorry. Oh, I think I need to share from here. I need to stop share this one and reshare that video.
Okay,
Just a
so
second.
not a problem. Anything you can try at your end, whichever works. Maybe then you can just pull out and again reshare your original screen.
Sorry. Is it visible?
Yes, now it is visible.
The screen is visible, no?
Yes.
Okay, just a second. Just a second. So this is the cell. This is one kind of cells. Here I will show you how this giant plasma membrane can be generated from the cell and how it looks like.
Dr. Banerjee, can you see a square box in the right hand side? Is it
Sorry. Yeah. I am sorry. Extremely sorry.
Okay.
This is some advertisement is popping up.
Okay.
Is it visible now?
Yes.
So this is a 40 minutes event. We compress it in very short time period. Please have a look. You will find some first appearance of some arrows. See the green arrows. Please follow those arrows. Now you can see that there are formation of this kind of blades. I think the video is moving a little bit faster in Zoom platform. Let me run it one more time. Okay. Sorry. So please follow this one. There will be, is it visible now?
Yes, it is visible.
Okay. So please see this. So here is the nucleation point. Then you will find that these things are getting bigger over the time. So from one cell, you can get multiple such kind of vesicles, and these vesicles are being generated with the help of certain kind of chemicals. So these are the stuff that we generally call as a giant plasma membrane vesicle. Sorry. Is that slide visible now?
Slides, no, you have to share it again. It's only coming your video.
Okay, I'm not finding. Just a second. Is it?
Yes, sir. It has come.
Now it has come, no?
Yes.
Okay. Yeah. So on the basis of that, when we confirm this one, so this GPMP can be used as an drug delivery cargo. So then here we designed an hypothesis that is related to some part of my other work, that is tissue engineering and regenerative medicine. So we want to check whether this cell-derived GPMP can be used as a regenerative cargo or not. So that means whether it can deliver certain therapeutics that can help in a regeneration mode. So one very interesting thing in this regard we did, most of the time if you see, you find that people are trying to differentiate stem cells with the help of some different kind of say reagents or growth factors to one particular lineage. Now, when you look into that in vivo situation, it has been observed that and mesenchymal stem cells, as it is in one of the key regulator which secretes a lot of chemicals and growth factors and cytokines to make an environment regenerative. On the other hand, it receives certain feedback chemical signals from its neighboring cells so that to maintain its particular regenerative capacity. So here we try to explore that. What we thought, what about looking for an osteoblast derived membrane vesicles and check its potential on that therapeutic potential on the mesenchymal stem cell. In an in vivo bone remodeling, what happened that osteoblast interact with the mesenchymal stem cells through paracrine as well as juxtacrine signaling, and then this mesenchymal stem cells performs its role for the regeneration. So our idea was, if we generate some membrane vesicles from osteoblast, can we actually mimic this functional role in an acellular platform? So what we did for this purpose, we thought this hypothesis that the giant plasma membrane vesicles generated from osteoblast as an acellular packaged cargo can provide necessary chemical cues to direct the osteogenic differentiation of MSC in a biomimetic way. So to answer or to test this hypothesis, we segregate the whole problems into a couple of different questions. The first part was associated with, can GPMP be generated from the osteoblast? Then second question is, can we tune its production? Then third, can we vary the GPMP compositions? On the other hand, here we ask, if can GPMP generated from this osteoblast be fused with the HMSC? And after fusion, will this HMSC survive? And then what will there be osteogenic differentiation of HMSC? So these are the 6 questions we formulated at the beginning, and the rest of the slides I will show how we answer one by one and finally lead to a conclusion that that GPMB from one cells has a good bioactive potentials and can be used as a therapeutic cargo. Now for this purpose, we generated 3 different kind of GPMB, G1, G2, G3. So here, so you see that what we thought of generating these giant plasma membrane vesicles from these osteoblast cells, but we thought to manipulate the osteoblast prior to generate this GPMB. So one was without any pre-treatment that we considered as in G1. Next one is that we treated this one with cytochalasin D, which one is an actin destabilizer. We considered is G2. And the third one we treated with methyl beta cyclodextrin, and we considered it as in G3. So we generated GPMB from this. This is a detailed protocols. I don't think anybody is interested in the protocol now. If you are, then please let me know at the end of this presentation. So there are 3 different kinds. So what is the purpose? Now one is the GPMB which have a native membrane composition. The second one have a GPMB where the actins are destabilized, and the third one a GPMB where cholesterols are sequestered. So in this way, 3 different kind of GPMB we generated from the osteoblast. Now here you can see that the fluorescence microscopic image, this is a time-lapse image of this GPMB generation, 10 minutes, 30 minutes, and 50 minutes. Now here you can see this is the over the time period how this giant plasma membrane vesicles are generated. So then what we check, what is the average diameters of this GPMB? And here we find that this GPMB diameter at the beginning, all 3 different kind of cells at the 10 minutes, there is not much difference in the GPMB generated in their diameter. But the moment we go a little, the time get increase, we find that there isn't started coming some differences. And then we find that the native GPMB generated from the cells with the native state has the biggest size in comparison to the 2 other. And simultaneously, we also check what is the number of GPMB synthesized per 10 to the power 5 cells. It's an effort to quantify these GPMBs. But one thing I would like to mention that the GPMB, whatever we are talking about, we focus on the cell membrane vesicles which are on the diameter of 1 to 15. There are some small proportions of vesicles which have a diameter below that. Now, this one very important question that may comes at this particular point, whatever you are talking about this GPMB, your diameter of the membrane vesicles is very close to that apoptotic bodies. So how will you ensure that your GPMB is not the apoptotic body? So for that, what we did, here are the 2 sets. This, please follow this vertical column. The first one is a positive control and second one is G1, that is the native state. So what we did, we first stain the membrane with the vibrant DIO, which is a membrane lipid binding dyes. And then we, in one set of cells, we treat them with the H2O2, which is an apoptosis inducer, and the rest, another one we did not. And after this treatment, what we did, we use annexin VP, so which is an apoptosis marker. So then what we see that in case of this positive control, where the cells were treated with H2O2 as well as this H2O2, then we found that here the bleb is containing this annexin V. So that means whatever the bleb is generated here, this is an apoptotic bleb. That's why it is having that same, it is taking both the colors or both the dyes. But when we treat the cells with the GPMB generating solutions, then we find that the GPMBs are generated, these green lines or green blebs, but those blebs are not having this red one. So it clearly suggests that the GPMB, whatever being generated from the cells, are not having this, are not apoptotic blebs in nature. Now we also check that whether this GPMB being generated, we confirm that they contains this RNA and other RNA and say proteins. What about the cytosols? How much cytosols are there? So what we did prior to generating this GPMB, we label the cells with calcinam and then we let the GPMB to form. And what happened that as the calcinamide is in dye for the cytosols, we found that all the GPMB is containing this particular green fluorescent dye, which clearly suggests that when the GPMB are forms, there is a transportation of cytosols from the cells to the GPMB. So this fluid membrane is basically, the fluid content is basically the cytosol. Then we check another way whether this GPMB, whatever been generated, are leaky in nature or not. So to test that, what we did, we synthesized these membrane vesicles in presence of an FITC dextran. So our idea was we mix the FITC dextran in the GPMB generating solution. Our idea was if the GPMB is leaky in nature, then the FITC dextran should enter into the GPMB. So when we'll take out the solution, after the synthesis, GPMB should appear as green. But we found that the GPMB is not appearing green. So at least we can say in a way that for a molecule FITC dextran of close to 12 kD molecular weight, so this is impermeable, this GPMB, which indicates its corresponding membrane integrity. Then we check this composition analysis. We check through NMR. So here we found that we compared this NMR data with the native GPMB, and we found that our native cell membrane, and we found that the G1 has a very close resemblance with the native cell membrane, whereas in both G2 and G3, their composition or all the components are there, but there are certain variations. This calculation I have not shown here, but on the basis of this NMR data, we'll calculate it that there are certain variations in the phospholipid compositions, although all the phospholipids are present, but the composition varies. And then we check the phosphorus per unit of cholesterol to check the phospholipids to normal lipid ratio. And there we find a very interesting thing that the relative phosphorus concentration is increased in case of G3. Because here you are taking out the cholesterol. So as your cholesterol has been taken out, so technically the ratio of phosphorus to cholesterol will definitely increase. So that happens there. Now our next job was to find out, as we are willing to check whether this GPMB can be used as a therapeutic cargo or not. So we thought to do one thing, that say let's generate GPMB from one cell, that is our osteoblast, and try to fuse it with another cells. So for this purpose, what we did, we stain our GPMB producer cells green, a membrane dye, and then we stain our HMSC with another dye red, and then let them fuse. So idea was that if the fusion is taking place, so we are going to get some cells where the both green and red will coexist. And if that is there, then we can say that whatever the content GPMB was having, that can be delivered, or that is being delivered to this particular mesenchymal stem cell. So here is the experiments with that. So here you can see these red are the HMSC, which are stained with this cytotracker red. And then we allow this DIO of stained GPMB. And then we see that there are a couple of cells which actually taken this become yellow. So this clearly suggests that there are a few which are only green, which are only red, but many of them are actually yellow. So here it appears a little bit orangish. So, but if you look into clearly, if you find that this intensity and the frequency of such an orange cells is not same for all the cases. So there are different kind of distributions. So then we'll try to quantify it through facts. And then when we quantify it, we found that the second population, which is purely this double stained population, in one case it is 20.2%, 19.93% and 29.10%. So there is a certain distribution. So the next graph, this is actually a typical one, which suggests about how this GPMB get or interacted with the system. So this particular experiment is known as a quenching dequenching experiment. I don't want to go into the details of this experiment, but in short, this suggests that if there is a fusion, there will be certain kind of decrease in this one, so that intensity, and that way we check this particular fusion environment. And then after this, we want to observe, once this kind of fusion been done, the few cells are still being alive or not. So we found that all the few cells are still added. Then once we, after fusion, once we let them adhere, they are adhering and they are looking normals and healthy, no such blebs. So this is a good indication that the GPMB can be fused with these stem cells. And then we check their live cell populations and their morphology. And finally, then after this post fusion, we check how this particular, once we fuse this osteoblast derived GPMB to HMSC, how it induce that HMSC to take an differentiating route. So here we find that the ALP productions of different kind. What is important here, you see that all the GPMB generated from this osteoblast are not equally potent to induce this alkaline phosphatase production in HMSC. That is to differentiate the cells. So here we found that the G2, that is where this GPMB generated from an osteoblast with an actin destabilizer, that can induce better alkaline phosphatase production. And similarly, we try to check the collagen type 1 expression. Then we analyze the post-fusion 14 days differentiation of HMSC by reverse transcriptase PCR for different kind of differentiation markers. And you see that there are different, in all the cases, there is an increased differentiation and with respect to the control. But among the three different kind of GPMB, there is some variation. Then we reconfirm the same things with the help of a qPCR, and finally we go for an long-term analysis of this. With this, when we allow these things for 28 days, these few cells, and check what kind of mineralization happening there. So here also we notice a very good kind of mineralization. So altogether, there we summarize that the GPMB can be synthesized from the osteoblast by using this chemical vesiculant, and they can be generated in presence of blockers, and they showed notable differences in production kinetics and compositions, and they can be fused with HMSC, and GPMB can induce osteogenic differentiation in HMSC. So with this, I would like to end here. So here are the students who contributed to this work. Dr. Jamini Jogalakshmi, she was my earlier MTech. So she completed her PhD in the last month from Institute of Pasteur. So she initiated this work in my lab when I was at NIT Rourkela. Then Ms. Dipanjali Gaur, she was in another MTech. So I'm proud that both of them were actually gold medalists of the department. And now Dipti is working on this topic. So we have a plan to check this kind of therapeutic applications of GPMBs in liver regeneration as well as chronic wound healing. She's working on that, and she's a recipient of this Indo-US Women Scientist Fellowship. Just this year, she visited US and a very good lab for working this. And here are my collaborators. So Professor Spas in Germany, Professor T.K. Maiti, my guide, Professor Shamir Kumar Pal, Professor Mohammad Salim. So he was earlier in NIT Rourkela, who was a very, integrated part of this. Basically, we two together started working on this. So he was an integrated part of this particular project, and we have research, and we have a joint publication, Dr. Sanjay Malik, Dr. A. Chanda, Professor A. S. Giri, and Professor Kunal Pal. With this, thank you. Thanks for your patience.
Thank you, Dr. Banerjee. So it's a very nice talk, and it's very informative, sir. So very wide, lot of data. And this the discussion will be open, and in the Zoom platform, anybody wants to ask directly can ask.
Sir, Indranil sir, can you just put down your slides, take back your slides from sharing?
Sure. Just a second. Is it
Hi,
okay?
Professor Indranil. Can you
Hi.
hear me?
Yeah.
I have one question.
Please. First of all, Sasmita, how are you?
I'm fine. How are you?
I'm fine too.
How do you extract these exoderms? Oh, sorry, your GM, not
GPMB.
giant. Yes,
So GPMB, we have a benefit that these GPMBs are little bit larger in size. So a normal centrifugations is good enough to, so there are differential centrifugations. So we do these 2-3 different kind of differential centrifugation. So in one way, we first centrifuge the cells and then we take the GPMB.
Is it, I mean, purely gives your GPMB?
So that is a bit tricky when you are doing this one. So the layer you are collecting, that is very, very important. So it depends a little bit on that hands-on that scholar who is dealing with. So
Okay.
which layer he is taking. So if he is a little bit, say inexperienced, you will often get a cellular contamination. That problem is often there. So that is there. So it depends on how effectively you can take it from the upper layer.
And what is normally the distribution, size distribution?
So normal size distribution, I think I showed a graph probably, size distribution. Or you are asking that altogether size distribution.
Altogether.
Altogether size distribution is most of the cases we get something 0.5 to what is visible in general. So because if it is in below that, we often won't be able to see. There are some that could be a debris or something, but so far it is visible is something 0.5 to 0.6 micrometer to 10, up to 10 we got.
With a PDI, have you ever checked the dynamic distribution?
Yeah, we check that dynamic distributions. But the problem is that with the PDI, we can't go for this. Actually, DLS cannot be used for this. The reason is, so you have a mixed population of one is in a micrometer range and another one is a nanometer range. So
Okay.
if you go for most of the instrument, if you look, either they are good up to 3 micrometer upper range, or it starts from 5 to above. So in a single particle size analyzer, which can give me an overall range starting from say 100 nanometer or 200 nanometer to 10 micrometer, that kind of instruments we don't have. Anyway, so it is in a one go checking is difficult. So what we did, we actually check it in a 2 different segments. One segment is in the micrometer level and another segment is a DL, in that DLS level.
Okay, thank you.
So if I have a permission, then can I
Yes,
ask?
please go ahead.
So we have some more questions, but I'll start with mine. The first question is actually taken by Sasmita madam. So I'll like to ask, like, how do you characterize, like, is it the surface markers which you target, probably CD markers, CD surface markers, CD81, CD9, I guess.
Okay. So Professor Bishmita, we have not, for the GPMB, whatever the surface markers are there, GPMB contains that, we have not, the surface profiling we have not done. Okay, so my student who went, I told you the Deepthi, she went to US with that idea. Our project was on that to go for this membrane proteomics. We thought to go for that complete membrane proteomics, but because of this COVID-19 situation, that work got hampered. So this overall protein profiling has not been done.
So how do you characterize? How do you, so you go by only size? Because as far as I know,
we go by, if you look into the presentations, we go for 2-3 different way. One is the size. Then we check for that your lipid compositions with the help of NMR. We check the lipid compositions with NMR of this particular. Then we characterize some kind of biochemical assay to check the corresponding phosphorus content, to check the phospholipid to get an idea about the phospholipids. Then we did some flow cytometry analysis to have a comparative idea about the granularity of the system. Then we try to check whatever this, as I showed that, whether it's contained the cytosolic components or this RNA or DNA, this kind. But even if you ask me individually what is there, so this kind of overall omics study, which can give me a clear profile, what is the component, that we have not been able to.
So I think with flow cytometry also,
majorly the reason is the budget, frankly speaking.
Yes,
So
I know, because even for flow cytometry characterization, you need literally high amount of antibodies, which are really very, very costly. I believe you can also characterize with flow cytometry. That is
no. Our point is that even if you go for an antibody, because we did not give thought to antibody based study, because antibody we will be only using when we have a specific application in mind. So whether this particular GPMB is containing this particular marker or not, or CD or not. But at this moment, we are really concerned to know what is the global protein profile of this. Not a single CD or single surface receptor, not like that. We first want to know the global profile. And for the global profile, our key aim is to go for this either microarray or complete NGS or complete, say, proteomics, something like that.
So from drug delivery perspectives, when it comes to the application, so have you seen, definitely you have results which automatically is already suggested, already proved that it is showing a better results. In case of osteoblast cells therapy you have seen. So have you applied into real-time osteogenesis?
No, that is the thing we are telling, that we are now, what we are doing this thing, we are now working on that another, the similar way. Here we first prove that this can be used as a cargo therapeutic. Now our next job is that now we are working on HMSC derived GPMV. And then we are checking whether this HMSC derived GPMV is good for, say, liver regeneration as well as this chronic wound. So this work are still going on. We are not in a very definite condition, but these are that now going on. And in the time, I would like to mention one very important point in this regard. So these therapeutic applications, what we are talking about, our group was the first who reported its therapeutic application. So later phase 1 or 2 papers.
Maybe in India, I don't think it's worldwide. It's already reported, but I think there's therapeutic effects already for liver generation, heart generation.
No, not with the GPMB, not with GPMB.
Not with the GPMB.
Not with GPMB. Exosome related lot of work you will find. Exosome microvesicle related lot of work you will find. But this GPMB was the first one which was a suggestive about that. And now people have started coming with lot of this similar kind of work.
Okay. So my point is, if the size is going to be large enough, more than 200 or 300 nanometers, or micrometer in size, so probably a drug cargo might not enter into the cell.
No, so our approach in this regard, you are absolutely correct. So most of the cases, if you look into this particular approach of membrane vesicles, people try to exploit the endocytotic route.
Yes.
Okay. Yes. But here we did not attempt the endocytic route because we know that this kind of size is not good enough for the endocytosis.
Okay.
Cell can't take, that's an obvious. But here, that's why we did another thing, because as this is a membrane vesicle, so there is a possibility it can get fused with the cell membrane.
Yes.
Okay. So our approach was to exploit that fusion property. And that's why we try to see if we change at the time of generating the membrane from the cells, if we can manipulate the membrane property a little more, can we actually increase the fusion?
So one more thing, when we say always talk about exosomes are a kind of replica to that of viruses or nanoparticles. It's a similar kind of thing with size range. So we always say exosomes are a better delivery system if you can control it, because these are cell-free. And when it comes to the stem cells, now the graft versus host rejection is much more. So when you are fusing the stem cells along with the exosomes, this membrane lipidic molecules as vesicles. So have you seen while applying, is there any kind of host rejection things happening?
We have not, first of all, we have not tried anything in that in vivo because we have not tried there. But this is a very interesting possibility, because this kind of fusion actually allows you to not only deliver the cargo, because if you look into this, that GPMB's overall membrane component or that quantity a GPMB is carrying, the membrane lipid, It is not an, it is quite significant. So if such an GPMB get fused with a cell, so it has a potential to immediately change the cellular membrane profile to certain extent. So there is a possibility that exploiting this, you can tune that cell membrane property as well.
So I don't think totally it will be lost. Maybe the fusion will take place and it will bring about something new characterization onto the cell surface, which is
exactly. So you can get some new features added to the membrane.
Yes. So I think with YouTube, we have one or two questions we have discussed about, and the discussion can go on. So just I'll take one or two few questions. Prabhat Kumar Swami, he is asking like, what is the maximum period of fusion can be done for GPMB in human stem cells?
Maximum fusion means?
Fusion period, like for like maybe how much time you are allowing you to fuse and give you a fusion this thing.
So here most of the experiments we did, so our protocol for fusion was that we take the cells, okay, and the GPMB, and we give them an, say a short spin for say 5 minutes, and then we let that together, we replate the cells and allow them to adhere there for 24 hours. After that, we start carrying any kind of checking. So, but if you ask me what is the actual time period required for this, there are certain kind of scope is there for the optimization.
Okay. So one more question, he's asking why these vesicles are actually vesicles or exosome vesicles are actually heterogeneous in nature. And he just wants you to throw some light on it because on its heterogeneous nature, because so probably you can.
Is he talking about the exosomes or the GPMB?
No, exosomes. He writes EVs. So basically, it's the exosome vesicles, exosomal vesicles.
So, extracellular vesicles. Now, the extracellular vesicles. So, if you look into the synthetic root of the extracellular vesicles inside, or I would rather say the biosynthetic root of extracellular vesicles. So, biosynthetic root itself is not a non-specific root, as it is a specific root. So, there is an obvious. It is quite obvious that it will become heterogeneous, because it is coming through following certain specific route. So there is no question that it will be an homogeneous system. Okay, so because it is generated from, if we are talking about that exosomes, it is started being generated from the endosomal route, and then it is following this particular track vesicles, and then say meeting with that inner part of the membrane, and then being throw out from the cells at all these phases. If you look into its interactions is basically heterogeneous in nature. So there isn't quite obvious that it would be heterogeneous.
So one last question in my mind is coming. So once the fusion with the stem cells and the vesicles, giant vesicles is happening. So is it necessary, do you think there is a requirement of going for genetic profiling, like genetic sequencing and checking it? Because we know that every step we need to again check all the parameters, whether right or wrong. So after fusion, there is any...
Yes, obviously, because if you look into this, what we are actually very excited about is that it contains RNA. Okay, yes, as it contains RNA, so we are very sure that it is not only containing the full-length RNA, it will definitely contain the fragmented and microRNAs. And if that is there, so there is obviously some certain possibilities that once you fuse these things, there you get some kind of inhibitory. So these RNAs can alter the genetic profile after the fusions, these small microRNAs. So it is very obvious to check. And we are, for the time being, so I would like to suggest that one thing, as I said that this group, our group started this work fast. So the point is, our objective is to, the work is already, it's in infancy. Yes. So we just got a proof that this can be exploited for therapeutic purpose. To what extent? Because that's why we are very, very keen to do all this omics analysis, starting from this proteomics, and that is not only the GPMB, it's before stage from
Yes,
where the
before
cells
stage
are
and
coming,
after stage.
at the GPMB level, what it contains, and after fusion, how it is changing. So all these three stage, unless we are doing this complete omics profiling, we are not in a positions to tell anything. Even I will suggest, I will tell that is a very, very preliminary requirement to go forward with this kind of research. But as you know, we all know that these are very, very budget const. We work under Beuria.
So actually, I believe there will be a
Tight budget.
time. Yes, so we don't have to actually send our students to US or any other laboratories outside India. Probably we will develop our own setups so that we'll do
Exactly,
complete
exactly.
profiling. Yes. So any other thing you would like to add, or maybe I'll just ask if anyone else is interested to directly interact with you. Probably from my side, I'm done with my questions. And
Thank
probably
you.
we have already taken questions from YouTube. Dr. Vinod Bihari can actually
So
say.
anyone from this Zoom platform want to discuss or ask question is free to ask. All right, good. So maybe there is no question from Zoom platform, but if anybody has any question, so they are free to email us, and we'll be also contacting the speaker, and we'll be in touch to answer all those questions. If any will be arising in the latter time point also, if not now. Okay, and with this we thank Dr. Banerjee for his nice talk and nicely articulated. So a lot of data, and although it is at infancy, it is a long way to go. So and it will surely success. And we are really thankful, sir, for your time. So
Thank you, Vinod sir.
Thank you, Dr. Banerjee. It was really nice being with you, listening to you, interacting with you. I cannot thank you enough for being here. So it's
You're most welcome, and it's a great experience for me, and I really enjoyed this platform and talking to you all. And thanks for inviting me once again.
thank you so much.
thank you, sir. Thank you, sir.
Okay.
So with this, then we move ahead with the session, and the last speaker is from our NIT Rourkela. So I think so Mohapatra madam, Sasmita madam is there.
Sasmita madam, are you there? Okay.
Yeah. So yeah, she's here. So let me introduce to the common audience. Although we know her, so I need to provide the introduction before that. So Dr. Sasmita Mohapatra has received her bachelor degree with honors in chemistry from Ravenshaw College, Odisha in 1999. She did her masters from chemistry from Utkal University in 2001. She then obtained her PhD degree from IIT Kharagpur in 2007. After that, she
Vinod sir. Vinod sir.
and am I audible?
Yes, now
actually
you're
we got
audible.
yes, now you are audible, but there was a pause for like 2 minutes. No, not a minute, but like 15 seconds or half a minute.
Okay, maybe I can start. So actually, see, Dr. Mohapatra obtained her PhD from IIT Kharagpur in 2007. After that, she joined the NIT Rourkela in 2008, and she is working as associate professor in Department of Chemistry, NIT Rourkela. She has published 47 papers in different reputed international journals with a total citation of more than 3,000 and an H index of 25, and she has been awarded with Young Scientist Award from Odisha Chemical Society in 2013 and Young Scientist Award from State Science Academy in 2014. And her current research interest is driven towards design of fluorescent nanomaterial for analytical and bioanalytical application. And with her lot of experience, so today let's listen from her details on the topic carbon nanomaterials for theranostic application. So and all the audience will be actually listening to hers, and in the due course of time maybe I can again remind that please post question as we go along. So if anybody keep forgetting questions or so, or else in the Zoom platform event we can discuss face to face. Okay, so Dr. Mohapatra, please.
Am I audible?
Yes.
Yes, ma'am.
Thank you, Dr. Sahu, for this nice introduction. First of all, I would like to thank Professor Nayak for inviting me to this platform. I'm sharing my screen. Just tell me whether it is visible or not.
Not yet.
Now?
Yeah. So it's started. So full screen and we proceed.
Can you see the full screen?
Yes.
Yes.
Okay. So the topic of my presentation is carbon nanomaterials for theranostic applications. Here I can outline my work, my presentation. First of all, I will give a brief understanding on theranostics. What is theranostics and why carbon are very special for theranostic applications. Then I will focus some recent work from our laboratory. What nanomedicine essentially means? Nanomedicine means it is to apply nanotechnology tool in medicine. Nanomedicine includes nanoparticles or nanoanalytical techniques in medicine. Now, what is the difference between nanomedicine and conventional molecular medicine? In case of nanomedicine, the diffusion, intermixing, and sensoric response becomes ultra fast by directly probing local properties in the nanodrop. Physical and chemical processes can be controlled, intensified, so precision is enhanced. And there is direct access to biomarkers, which is not possible in case of conventional molecular medicine. That imparts the nanomedicine with a site-specific delivery. Then real-time monitoring becomes possible in case of nanomedicine. With these concepts, a research in the intersection of broadly true areas in nanotechnology and molecular medicine that may produce a route to personalized medicine. So in the next slide, I will show when this nanomedicine was conceptualized in 2003, people focused on two basic concepts. First of all, how to enhance the therapeutic efficacy of existing drugs, because existing drugs are mostly these are hydrophobic in nature, so these are not good for prolonged circulation in the body. That's why if you can increase the solubility of these drugs by encapsulating in some nanoparticles, then it can be circulate in the body for a prolonged time. Next is specific targeted approaches, that is site specific delivery of the drug, because when normally in the conventional way what drugs we take, that is distributed throughout the body, in that case there is some side effect which is observed. But in case of specific approach, we can introduce some ligand with high binding affinity towards overexpressed or highly specific towards highly specific receptors. That means we can encourage the site specific delivery of the drug in order to reduce the side effects. But the present state of art in case of nanomedicine that emphasize on nanotheranostics. Using this conventional two approach, we have also done some work in our early research from our group. But the present state of art in case of nanomedicine that gives emphasis on development of nanotheranostics. Theranostics is a word which is generated from combination of therapy and diagnosis. So what happens in case of theranostic medicine? There is a in case of theranostic medicine, a single drug can solve multiple purposes by loading different therapeutic cargoes in the drug. For example, if you take a nanocell, you can load multiple therapeutic agents, as I have shown here, therapeutic agent 1. We can decorate with a molecular marker, even we can decorate with different molecular markers. Then we can decorate with a hydrophilic spacer, which will minimize the possibility of opsonization. We can decorate with a fluorophobe. So multiple facilities can be incorporated in a theranostic agent to solve different purposes, which is not possible in case of conventional medicine. Now, why carbon materials are so special for theranostic applications? Just for your view, I am just telling there are various carbon allotropes available till now, like diamond, graphite, fullerene, graphene, carbon nanotube, graphene oxide, and this is an emerging carbon allotrope, that is carbon dot, or in many citations it has been reported as carbon quantum dot. So in these allotropes, this hybridization of carbon that varies from sp to sp2 to sp3, depending on the hybridization carbon, the properties of these allotropes varies. Now, why for theranostic application, the properties of carbon nanomaterials, such as inherent optical property, that is very important for diagnostic imaging. Specifically, the light absorbance at NIR region, that is the higher wavelength region, 750 to 1400 nanometer, is good for photothermal and photodynamic effects. Then NIR has greater penetration depth, lower excitation scattering, and autofluorescence. Because if once autofluorescence is a process which is disturbing in case of monitoring the biological processes, mostly the water as well as the biological tissues that absorb in the visible or UV region, what is also the traditional dyes what you use in observing the biological processes, that also absorb in the UV visible region. So that disturbs in the monitoring the biological processes. That's why carbon nanomaterials that absorbs in a higher wavelength region, the autofluorescence from biological tissues or water, normally that does not interfere in case of carbon nanomaterials. So that's why it is a very good diagnostic imaging agent. sp2 carbon materials have a large surface area. That's why it is a good substrate for loading aromatic drugs. Mostly the drugs are aromatic. I'm just telling from chemistry point of view. I think it will be, it is understood to a large audience. sp2 carbon materials has a high surface area. Can you see the screen?
Yes,
Yes.
it is. We can see.
Yes, sp2 carbon materials have high large surface area. That's why it is a good substrate for absorbing aromatic drugs. That is one aspect of carbon materials. And lastly, several chemical modification strategies can be used to conjugate various moieties to generate a diverse array of functions. For example, you can conjugate a ligand, you can conjugate a marker molecule, you can conjugate a drug on carbon nanomaterials. So it has versatile roles. Cancer multi-drug resistance, and that is a, you can say this is a phenomena which leads to therapeutic failure in case of chemotherapy. In a traditional chemotherapy, cancer multi-drug resistance is a phenomena which leads to failure. In order to overcome this MDR, there are many inorganic nanocarriers have been reported to overcome this. If you see this data from 97 to 2015, the publication on application of nanocarbon based drug carriers for carrying anti-cancer drugs, that is exponentially it is increasing. So with this background, now I will show some of our work driven in this direction. As I have shown, carbon quantum dot is it is an emerging carbon nanomaterial. You are acquainted with graphene quantum dot. So what is the basic difference between carbon quantum dot and graphene quantum dot? Graphene quantum dot is a extended sp2 network So it is a highly crystalline structure, whereas carbon quantum dot that consists of a crystalline core and an amorphous shell. So broadly, you can understand that the core is sp2 and crystalline, and the outside shell is amorphous, and it is the outside shell which imparts excellent luminescence property to carbon quantum dots. A quantum dot of a quantum dot can emit multiple colors depending on the excitation wavelength. So what is the advantage of this? Suppose you want to monitor some biological phenomena like the release of the drug or interaction of the drug with the cell, then what you can do, you can tag your drug delivery system with this carbon quantum dot, and under different excitation wavelength, you can see the movement of the particle or drug. So it is an very much advantageous in case of real-time monitoring of the therapeutic response. I'm telling I'm a chemist. From day one, I'm a chemist. So we normally focus on the synthesis of the material and the application of the material. So with this excellent properties of carbon dots, we tried to synthesize carbon dots from edible orange. We extracted juice from the orange. Then by hydrothermal treatment, at comparatively low temperature, we are able to produce very highly fluorescent carbon nanoparticles. You can see in this slide, there is this cuvette which is showing yellow under daylight that appears very bright green under UV light. So if you can compare the fluorescence of FITC with this carbon quantum dots fluorescence. And the most interesting fact about this is if you go on increasing the excitation wavelength, it emits different colors. Particularly, this property is based on the different surface states between the HOMO and LOMO. I need not explain this thing here. Let us see the application side. Then what we did, we have verified the cytotoxicity of this quantum dot, and we found that this is tolerable up to 200 microgram even after 72 hours incubation. That was done in Mg63 cell, and as you can see here, the interesting fact is that when we excite it under 488 nanometer, bright green color was coming. When we changed our excitation wavelength, it was bright blue color. So this work was published in collaboration with Professor T.K. Maiti from IIT Kharagpur, and it was published in ChemCom. Next, with our interest to develop a multifunctional nanoparticle, multifunctional means to develop a theranostic particle by combining optical imaging, targeted drug delivery, imaging, that is magnetic resonance imaging. In a single particle, we try to incorporate the quantum dot which we have synthesized from orange juice in a hollow mesoporous structure, because hollow mesoporous structure is an excellent material for drug delivery. Because of its large surface area, it can hold a large amount of drugs, and the surface can be easily modified. The gas loading capability is quite high, so it is an excellent material. What we did with hollow mesoporous silica, we deposited this orange juice by some chemical method. Then this material is fluorescent. In the core there was some magnetic particle. On the surface there is fluorescent quantum dot. So overall we developed a theranostic particle combining the magnetic properties, then high drug loading capacity of mesoporous silica, then fluorescent properties of quantum dot. Then how we applied it in theranostic application, that is another story. This is a drug called camptothecin. That is a plant alkaloid isolated from Camptothecica acuminate. Normally the Japanese people, they use this leaf of this plant and they in their tea. This camptothecin shows significant anti-tumor activity in a broad spectrum of human malignancies. How this CPT works, it inhibits the relaxation process of DNA related to the replication and transcription by stable combination with topoisomerase and DNA. But in spite of this crucial role, the application of camptothecin is limited because it is not soluble in water. So what we did, we tried to incorporate camptothecin in the particle, this luminescent magnetic particle, what we have synthesized in a chemical conjugation route. How? This is the structure of camptothecin, what we have shown here, but it is not water soluble. We tried to increase its water solubility by conjugating a water soluble chain, as you can see in entry 6. So this conjugate of camptothecin is soluble in water. Then we conjugated on the surface of this particle by chemical conjugation. So overall, this particle, it was magnetic, it is fluorescent, and now it is containing camptothecin. Further, we attached a folic acid on the surface of this particle by chemical conjugation technique. Now, this folic acid will direct this theranostic to the folate receptor, and camptothecin will be released on the application of stimuli. That we studied in the drug release profile of this, and we observed that camptothecin, the release of camptothecin was highest at that was highest at pH 6 point, I think 7.6 in presence of an enzyme called cathepsin. It was observed that almost 90% drug was released from this theranostic particle. When we studied the cytotoxicity, we found that the IC50 value of the drug loaded particle is 2.1 microgram per ml, which is much lower than the free drug. Then we applied it, and you in the cell, in the HeLa cells, I think. So this theranostic particle induced cytotoxic effect in the HeLa cell, and the nuclear apoptosis was found with 10 microgram per ml of the nano drug. Because of the presence of the magnetic core, this is a good MRI contrast agent. This work we have done in IGH Rourkela. When we R2 is called relaxivity, transverse relaxivity. That is a parameter which is used to show the MRI ability of the reagent. If you compare the R2 relaxation time, transverse relaxation time of our drug with commercially available MR contrast agents, we found that it is much better than the commercially available Feridex, Combidex, like that. These are the drugs which are marketed by different pharma companies. Feridex is a MR contrast agent, that is a dextran coated iron oxide. Combidex is also an iron oxide based MR contrast agent, and we have compared it with our drug, and we found that it is much better. It has 150 R2. This cell killing ability, all this work has been done in Professor S.K. Bhutia in the Department of Life Science, NIT Rourkela. So now I am showing a quantum dot which is targeted at certain cancer receptor. This is called Cialin-Lewis A. This is a oligosaccharide antigens cancer marker that is mostly overexpressed in liver cancer cells. Even in also other cancer cells, it is there are some reports, but mostly it is overexpressed in liver cancer cells. So in order to target this marker, we have developed a quantum dot. Before developing a quantum dot, we have to study the chemistry of this cancer marker. We found that there are cis diol groups which are present in this Cialin-Lewis A. So keeping it in mind, we have developed a quantum dot which is having boronic acid groups on its surface. We have synthesized it by hydrothermally decomposing thiourea, 3-aminophenyl boronic acid, and citric acid. The interesting fact about carbon quantum dots is that since it is synthesized at comparatively lower temperature, the structural memory of the precursor, for example, we are using here 3-phenyl boronic acid, that is retained on the surface of the quantum dot which is produced. Now we have isolated this by different techniques. Then we found that this quantum dot emits bright luminescence at lambda at 520 nanometer. When excited at visible region for 20 nanometer, that exists bright, that emits bright luminescence. And because of the presence of boronic acid groups on the surface, it has affinity towards Cialy-Lewis A. It can bind, it can recognize Cialy-Lewis A. When you suppose we are taking it in a cuvette and it is bright green in color, when you add Cialy-Lewis A, there is significant increase in the luminescence of this quantum dot. So we have utilized that molecular recognition property of this quantum dot, and we used it to image liver cancer cells. It can differentiate normal cells from Cialy-Lewis A overexpressed cancer cells. In this picture, you can see there are two rows. The first row shows liver cancer cells, and this second row shows normal cells, L929. When we incubated with this boronic acid functionalized quantum dot and visualized under confocal microscope, the luminescence from liver cancer cells was very bright. That indicates that the quantum dot is selectively taken by liver cancer cells, and there is the brightness increases. There is another theory behind it. Here I am showing it. When quantum dot comes in contact with the receptor, sorry, yes, this receptor molecule Cialy-Lewis A, there is molecular recognition because of hydrogen bonding. That interaction of this uh, quantum dot with Cialy-Lewis A, that brings these quantum dots together, and their aggregation is induced in the quantum dot. This aggregation induces manifold increase in the luminescence intensity, for which it can be used as a fluorescence imaging agent. Again, what we did, okay, we have developed this one. We have used it for luminescence imaging. So our interest to develop a theranostic particle again dragged us to incorporate this quantum dot in a mesoporous silica surface. Mesoporous silica is a good substrate for loading drugs. Now in this work, we have incorporated both T1 and T2 contrast agent in the center of the mesoporous silica. Then one thing I can say here, mesoporous silica, there is a pore. This is a porous material, but porous material, the molecules can be diffused through these pores. But if you can put a cap on this mesoporous silica, the diffusion of drug molecules, that can be finely controlled. Suppose you can remove this cap under certain stimuli, and the cap is closed in the normal atmosphere, then what will happen? The drug cannot be, it will safeguard the drugs. When there is stimuli, then the drug will be released. Okay. So utilizing that, we have developed a stimuli responsive drug delivery system. You can see this is a TM image of the mesoporous silica. At the center is dark, which shows this MR contrast agent is in the center, that is a gadolinium iron oxide, and the outer shell is a mesoporous silica, and on the surface there is deposition of this boronic acid modified quantum dots. After you see the entry E, that it appears black. When the quantum dot is released under the stimuli, then this the pores are clearly visible. So in that way, we have judiciously we have constructed this stimuli responsive theranostic system, and we verified it in different cells. We have seen the drug, we have seen the cytotoxicity of this developed theranostic system. When it was as it is, when there is no drug, there is no change in the cell, no significant change in the cell viability. But when the drug was released under low pH means stimuli responsive. In presence of stimuli, there was drug was released and there is significant drop in the cell viability. That means it is nicely working for controlled drug release. We have also used it for imaging cells, and it induces nuclear apoptosis, which was which can be visible in this. By DAPP staining, we can visualize the nuclear fragmentation in this induced by this stimuli responsive system. And the work was done in collaboration with Cultivation ISCS Kolkata. Dr. Pramanik has in his with his assistance, we have done this work, and it was published in Langmuir. Now, the last part of my research work in this direction, that is mesoporous carbon nanospheres in photothermal therapy. What is photothermal therapy? Photothermal is a term which means how light can be converted to heat. When light is exposed, when some material is exposed to light, it generates temperature. And traditionally, gold nanorod has been used in cancer therapy as a photothermal agent. Gold nanorod, copper sulfide, all these are used. But the photothermal conversion efficiency is poor. Photothermal conversion efficiency is a parameter which shows how efficient is your photothermal material. You can see in this picture, if you inject this photothermal particle in a certain model, in vivo model, and expose it to laser light, then there will be a temperature of this the local temperature will be high, which will kill the cancer cell. Because beyond certain temperature, cancer cells cannot survive. For example, certain cancer cells cannot survive beyond 45 degree, like that. That varies from cancer types, cancer to cancer. If you expose this with laser light, then the particular area, that killing effect may be induced. That is called photothermal therapy. And another fact is that if the material is fluorescent under NIR laser, then simultaneously we can also see which area the killing effect is induced. And the carbon materials are excellent NIR absorbers. Normally, the materials which absorb in the NIR region are used as photothermal agents. Mesoporous carbon materials are excellent photothermal agents. What we have explored means our research group has recently explored this one. Mesoporous carbon particles are emerging as attractive photothermal material because in case mesoporous carbon is a material which is generated because of the self-assembly of tiny carbon nanoparticles. So in the intersection region, normally carbon absorbs in the visible region, but in the intersection region can absorb at NIR region, which is exclusively responsible for the temperature generation. I'm showing this carbon particles we have developed in our lab for that is a long synthesis process. We have optimized the synthesis of the spherical carbon nanoparticles below 100 nanometer, because the synthesis of carbon nanoparticles is really challenging. It is difficult to produce spherical carbon nanoparticles below 100 nanometer. Normally it goes beyond 200, 400 like that, but within a nano dimension spherical particle with uniform size is really challenging. We took a long time to prepare this particle, and these are also fluorescent. We have seen under visible as well as NIR radiation that emits green fluorescence. We have measured the temperature, the rise in temperature with respect to concentration of these materials, with respect to time, and we have found that the photothermal conversion efficiency is 52%, which is much higher than what we get in case of gold or copper sulfide. Then we have also studied it in oral cancer cancer cells, that is FADU cells, and we have observed through facts that a reasonable fraction of cells are getting damaged by laser irradiation for a very a short period, I think 2 or 3 minutes, a reasonable fraction of, he can see here that 31%, 31 plus more than 50% gets killed when you expose this, after incubating these carbon particles with cells. If you expose under NIR laser for even for a short period, a huge amount of cells are getting damaged. This work was published in Langmuir. Recently, we have done this work in collaboration with Professor Sujit Bhutia in Life Science Department, NIT Rourkela. So with this, I conclude my talk for this webinar. The versatile nature, I can say that the versatile nature of carbon nanomaterials is ideally suited and expected to have a major role to tackle complexities associated with biomedical application. A productive collaboration between bio and nano, means nanotechnology and biology, that would facilitate this kind of research. So I invite technologists, scientists from biology group to collaborate with our group to produce some good work which will drive this kind of laboratory from laboratory to clinical stage. To take this kind of work from laboratory to clinical stage, a productive collaboration is very much essential. So at last, I would like to thank my students who have put their hand. Whatever work I have shown here, that is mostly done by my first batch of PhD students, Swagatika and Rahul. And I thank sponsor as funding agencies, DBT, DST Nanomission, and BRNets for supporting this, financially supporting this research work. Thank you, Sasmita madam, for inviting me to this seminar.
Thank you, madam. Thank you, madam, for such a nice presentation. I think it's over to Dr. Vinod. He will just, you can pull down your slides.
Yeah, Dr. Mohapatra, it's a nice presentation, elaborate so much of data. And this platform is now open for discussion. If any question, we can directly
can you see my
ask
slides?
question.
Slides
Yes, we can stop sharing.
Okay, okay.
From the bottom. In the
stop share.
where,
Okay.
Yeah.
Stop share.
Yeah, so anybody has any question, please, you can directly interact with Dr. Mohapatra. Zoom platform, anybody?
So
maybe now switch to YouTube. We are also YouTube live, so.
Okay, so in YouTube, I think 1 or 2 questions are there. I've just taken the screenshot to be just fast and easy. So Prabhat Kumar Swai is asking, what is the size of nanomaterials you are looking for your study and its effects?
Nanomaterials must be less than 100 nanometer, but very small size nanomaterials are also very toxic. A size between, you can say 100 to 120 or 130, that is best for biological, especially drug delivery applications.
Another question, it's like, what is the cause to carry out CDs for like 27 hours after incubation? You have taken the CD spectra and all. So I think that is being asked. What is the cause to carry out the CDs?
I could not follow. It is the protocol which is followed. Why it is incubated with cells for a long time? Or
no. What is the cause that you are taking the CDs for 72 hours of incubation? After incubation, probably the follow-up steps.
That is a normal protocol which is followed to, realize the interaction, whether CDs are going into cells or not. That is a normal protocol.
So my question will start with a generalized one, that why you have chosen orange juice and why not, what made you to choose orange
juice? That is also very judiciously chosen. I will tell you the entire story. The work was done in 2012, right? So in that, actually, I have given a scheme to my master students. At that time, carbon dot was very new. Can say our paper was the second paper which was published in for synthesis of carbon dots. It was very new. Only from sucrose and citric acid, it was published. Combination of laboratory chemicals, that is sucrose and citric acid, in a definite ratio, that was published to produce carbon dots. At that time we thought that why not orange juice, because all these precursors are available in the orange juice. Citric acid is a major component in the citrus fruit, and orange juice especially, that is rich in sugar. So if you take one precursor, then the citric acid which is available, that will self catalyze the process. No need to add the extra acid. So all these thoughts that implemented in that work, and we are quantitatively we are able to extract carbon dots from orange juice. And what is I think 400 milligram carbon dot was extracted at a time from that orange juice.
So if citric acid is the component, major component, I think if we are focusing on the citric acid, maybe
Sucrose.
we can also sucrose also. So maybe we can go for lemon juice and other things.
That is, after that you'll know. After that, a number of bioproducts, biomass was reported to produce carbon dot. People have, if you Google it, synthesis of carbon dot from biomass, you will get a number of precursors like onion, then lemon, pomegranate. All these carbon rich. Normally, bioprecursors are carbon rich. Okay, so that forms the basis of this type of work.
So supposedly, because you are a chemist, so my personal interest would to know, like, if you want to really enhance the photochemical activity or photothermal activity of a carbon nanotube or a magnetic nanoparticles, what would, like, you have also worked in toppings, and how you can enhance, what kind of materials you can choose, and
yes,
how can
first of all, first of all, you have to see the absorption property of your material. If it is absorbing in the NIR range, then only it will be, that is the basic requirement to be a photothermal agent. If your material is absorbing in the longer wavelength range, means after 750, if it is showing sufficient absorbance, then it will be a good photothermal agent. Next is how this photothermal agent can be introduced in the cell, that is the surface compatibility. You can incorporate in a hydrogel, or you can use certain kind of PEG PEG type derivatives to decorate the surface. You have to make the surface compatible so that it can be taken up inside the cell. So another point is that suppose it is showing absorption at certain NIR region, then particular laser you have to choose. Suppose it is showing at 808 or 840, then a particular laser of that wavelength only you have to expose.
But can we chemically enhance the absorption spectra of particular nanoparticles? Like,
yes,
I know there are set of nanoparticles which is being taken and conjugated with various kinds of fluorescent molecules, tagged fluorescent molecules, and those are commercially available for even biological purposes, like tagged to antibodies to stain them, what we generally use for our flow cytometric studies, and they are very, very costly, very, very costly.
Very costly.
Very costly. So if you can synthesize and characterize, so how we can change, we can modulate these nano dots.
See, in my material, since the material is itself fluorescent, so I don't need any kind of fluorophores, means organic fluorophores, to attach to the material. As I have shown you, I am developing quantum dots. As simply, I am just incorporating quantum dots in the drug delivery system. That quantum dot is working as a substitute of organic dyes, what means organic fluorophores. So in that way, the cost is very, very less.
So if we can,
without adding further stain, you can just see it where it is going.
Yes, sir. Yes.
So, but then what is the visualizing, monitoring? So how we will see it then? Like, will it autofluoresce?
Yes, it will show autofluorescence. Autofluorescence.
Okay.
So, and thing is that the fluorescence absorption, where it is absorbing, that is completely different from biological environment. In cell there are proteins, water, and other tissues which will perturb your absorption and emission property of the fluorophore, because those tissues also that absorb in the UV and visible region. If your fluorophore is absorbing in a complete different region, then you can correctly, your data is correct and you can correctly monitor the activity of your theranostic particle.
So you have seen where exactly it is going. It is on the surface.
Yes, that in some work actually we have seen it in under confocal, but right now your department is initiating the process to take a good confocal microscope. So I am I am emphasizing to put some source which will absorb in the longer wavelength region. NIR source.
Okay. So yes, after a long period of time, I think we are now initiating this process to have a confocal.
Yes, really, that will really facilitate this kind of work. So I am very much happy. It is a ray of hope for me, for my research.
This nanoparticles can travel through the plasma membrane of a cell, right?
Yes.
And can it travel through cell wall of a plant cell?
Cell wall. It will, when it is coming in contact with cell wall, normally it is taken up in by forming one endosome like that.
Endocyclic or cyclic.
Endocytosis.
Endocytosis.
But cell wall, there is it might travel through or not. I was.
It will travel through the cell wall and it will be taken up by the
Sir,
cell.
why don't you induce madam sir nanoparticles to your cells and we'll see under microscope?
Yes.
They are all.
Yeah, maybe we can check whether it can travel through plant, and if you.
Yes, very good. That will be very nice, because to monitor environmental changes or processes, there are some recent reports where you can see by tagging some nano particle, you can see the environmental change in your plant.
Right. So actually we have some compounds which are inhibiting a pathogen. So, and we want to put that one to the plant so that it will be get rid of the pathogen attack.
Okay.
And we are also not sure whether that chemical, if we apply just simply by liquid format, whether it will reach the destination and act on the pathogens. So that's why I asked.
In plants, actually, yes. Plants, I have never. I mean, I am not exposed to that area. I think can be done. That can be done.
Maybe I'll talk to you personally, and we can.
You are always welcome.
Yes,
I always welcome this collaboration of life science with chemistry. I always welcome.
So really,
that will be a productive collaboration.
I'm the only one who work on the plant in here.
Yes.
So maybe I can come personally.
At any time, you can come to me and discuss what is your requirement of nanoparticle.
Yeah. Yes. Okay. So thank you, madam. So anybody has any other question in the platform, Zoom, or any more question from the YouTube?
Let me. I'll just check. Just faster. I'll check. So I don't think we have further questions.
Okay.
So we can just.
Okay. So then, if we do not have more question, we have already a long discussion. So and still maybe a few more questions are still there, and even I am going to come up and talk to Mohapatra madam. So on my work even. So with this, then I thank Dr. Mohapatra for her nice presentation and a nice discussion, and quite plausible collaboration might happen. So then with this, I thank Dr. Mohapatra again, and also I thank all the three speakers for this session. And with this, I think so we came to a conclusion that this session will be over.
So I thank you, madam. Actually, when I joined, you were already there. So it's been long, but this is my first time that I'm listening to you directly, you presenting. So I'm glad that you are here and you.
I also hope that we'll have a productive collaboration in your future.
Yes.
So, okay, thank you very much for inviting me.
Thank you, madam. Vishwanta madam, so then I would like to request Vishwanta madam to please elaborate on the whole of this conference, this 2 days conference, as it has come to an end. So please take over.
Thank you, Vinod sir. Actually, this too, we always thought of having a grand conference in our department, but due to COVID situations, it's already 5 years, and you know that every 5 years we generally go for an international conference. This time it was not possible, but this was the time when we generally come up with a conference in every 5 years in life science department. But in any case, this interaction was very good for me. I have learned a lot from the speakers, and I would like to thank everyone from my behalf, our director sir, our HOD sir, Professor Patra, who has actually guided us for many things. So I'll just quickly summarize whatever I have seen from my eyes those 2 days. First of all, I would like to thank Dr. Amulya Kumar Panda. He is my guide, and I have done most of his my PhD work from National Institute of Technology. He's not my direct guide, but he's more than guide, I can say. He has helped a lot, and he is still there all along my career. So he's a great inspiration for not only for me, to all of us here. And as he's the director of National Institute of Immunology now. It was really very difficult for him to come and actually address a gathering, but as he himself told, he could not say no to me because I am his student. In any case, we know that he, despite many things, he gave us a real-time picture what's going to happen in case of a vaccine, in case of COVID-19. He has many experiences, patents which he described, and the drug delivery and the drug aspects, everything, the vaccine aspects, everything, whatever I have learned, it's majorly from his laboratory. So I cannot thank him enough for his contribution to science, as such, to humanity, to all of us. So he is always a learning person. Then I would like to thank all the speakers as well. So coming back to what we learned this 3 days program. So actually we started with the aim. If healthcare is our only purpose, for my thing, I would say that coming to the healthcare, and we always look for translational research. We do a lot of research, but unless it meets the needs of the patients and patient complaints, so I think it's not done. So we go through the needs of the patient. Then there are many aspects like what is need to be done, the drug molecules to be invented, and finally the regulations, how the drugs can be taken for further manufacturing, and then finally this drug can go and reach the need of a common man. So in that direction, I was pulled towards bringing people from academia and industry together, so that they will give us a real-time scenario if someone wants to be an entrepreneur or start their own things, or they have a product in their mind, how they can go for strategically, they can go for various steps like formulation part, from patent to the company manufacturing, and finally reaching to the common person. In that aspect, the marketing is also necessary. The manufacturing, the conversion of the product from bench to bedside, for this, it needs to be really monitored at various stages. That's why it was my aim to bring all the people together to listen each other. May not be every session would be interesting to everyone, because everybody's idea is different, but then putting together was my aim. I think with that, we have started our talk session, inauguration session, with Dr. Amulya Kumar Panda, followed by Dr. Veena Menon. She has a vast experience, and through her experience, she spoke about how to go about various kinds of like manufacturing processes at every step, the biosimilar products, if at all we don't have a product of our own, like we can design, manufacture various kinds of biosimilar products. Then they're monitoring the large scale production, and how the various kinds of clinical, non-clinical, clinical pharmacological studies can happen. And finally, we can take further for the market availability. And we have Venkat Ramana. He is with his huge experience. He's heading a company of his own. So he introduced us to various kinds of technologies and various kinds of diagnostic methods, various kinds of tools by which we can characterize and further go for it. Then Dr. Vipin Verma, he just added to the aspect like how the artificial intelligence can be helpful in drug designing, drug discovery. Like he talked about nanorobots and how things can change in next 10-15 years from now by particularly research in this area, and artificial intelligence can take over and can aid more to the drug delivery aspects. Then we have, in the today morning, we had Dr. Monalisa Mukherjee. We know she's heading the Department of Click Chemistry. She spoke a lot about the various kinds of hydrogels, various kinds of drug delivery systems in case of cancer and other infectious diseases. Dr. Ajit Kumar Mohanta, he talked about digital health ecosystem and how digitally we can control, monitor the various aspects of human health. We can connect the patients to the doctors, and we can monitor telemedicine, and we can monitor various kinds of aspects, and we can provide the right kind of solutions at the need. Then finally, Dr. Tusar Beuria and Dr. Indranil Banerjee, Dr. Sasmita madam. So all these lectures, they have given us enough insights towards drug delivery systems, and I think it would be definitely fruitful. Anybody who is looking and going to the YouTube links any time after this, and they can find the presentations and they can get benefited. So I hope it must have helped our the viewers, the participants who interacted, who asked questions, who were there all together these 4 sessions in these 2 days. For participants, I will say those who have attended the YouTube channel, and the feedback form will be provided to all of them, and after only we receive the filled feedback form from the participants, we will be giving you the participation certificate. So that's all from my side, I can say. And I think now I will ask Dr. Srinivasan if he's there. Dr. Srinivasan, are you there?
Thank you. Yeah, yes, ma'am.
So over to Professor Srinivasan. So he will be presenting the vote of thanks, and he will declare the session to be closed. So over to Professor Srini.
Yeah. Okay. Thank you, ma'am. So a very good evening to everyone. And the 2-day long sessions has came to an end, and I believe that the session has went very well, and it is a very successful event. And I'm privileged to present this vote of thanks at the end of this event. So I would take probably a minute of.
So Dr. Srini, a little bit louder. Dr. Srini, just
yeah. You couldn't hear.
I can hear, but in the interest of all, I'm just asking you to be a little bit louder or clear.
Okay, fine. I'll be louder. Yeah.
Yes.
So it's my pleasure to present this vote of thanks, and I would just probably take a minute or a couple of a minute to thank everyone who had contributed for the success of this event. And now coming on to the first speakers, we had speakers from academia as well as from the industry, and Professor Sasmita, she has elaborated the contributions and the lectures what they have presented. So first of all, I would like to thank all the speakers who had shared their precious knowledge with all of us and enlightening us in the field of drug delivery and molecular medicine. And now moving on to the others, like our first, the Honorable Director. So he has supported to make this event possible in this COVID time, and I would also thank other panel members of this conference, like Professor S.K. Patra and Professor S.K. Beuria, Professor Rohan Dhiman, Professor Vinod Bihari Sahu, and Professor Vidya for their efforts to conduct the event in a timely manner, and also the other faculty colleagues. Those are provided extended their support, as well as they have spared the time to conduct the event and for the encouragement. So we would like to thank all our faculty colleagues for this. And now it's to the participants without whom like the event wouldn't be a great success. So we thank you for paying your valuable attention in the conference and making the sessions very interactive with your questions. So we really thank you all for your participation. And there should be a special mention to Dr. Beuria Sahu and his team for facilitating the technical assistance toward this event. So without the technical support, I don't think like the event would be a very successful one. So a special thanks to them. And also to the Professor Sasmita Nayak for conducting the event for the entire 2 day. And the event wouldn't be successful without the support of students as well as the others, those who are behind this event, like indirectly those who are supported. So we thank you all so much for your efforts. And finally, I would like to thank everyone and especially to the speakers on this forum because they have for sharing their knowledge again. So thank you all very much. So and we request the all the participants surely to provide the feedback so that we may improve ourselves like if anything required, and your certificates will be provided as early as possible. So thank you all very much. And by this, we came to an end. Okay, thank you all.
Thank you, Dr. Srini, for such a thanking everyone. I should thank you for being with me, our team. So thank you, everyone. And for any feedback and for any other thing, you can always reach out to us and email us, and we'll be providing the certificates after getting the feedback form. So thank you all, and bye for now. I think we'll close the session and we have already come to an end. So thank you all. Thank you, Dr. Srinivasan. Thank you. Thank you. Bye-bye. Okay. Thank you so much.